Good morning, students. So, coming for the next topic, that is nitrogen cycle. So, what are all the contents which are being covered in this particular nitrogen cycle chapter, or the nitrogen uh, fixation? So, what are all the contents? Let us see first. With respect to this nitrogen cycle introduction, first we will be discussing. Next is nitrogen fixation. After that, non-biological nitrogen fixation. Among that nitrogen fixation, there are two types: biological and non-biological. And about the nitrogenous complex enzyme activity or the mechanism, and the symbiotic nitrogen fixation and the symbiotic under that. And energetics and regulation, I will be discussing later with respect to this energetics and regulations, and particular topic with respect to that nitrogen NIF genes, right? Coming for the introduction. So, what you all know, sorry, you all familiar about this nitrogen. What is this nitrogen? Which will be the particular component which is present in the, it is a very important essential component or the essential constituent of the biomolecules, which will be found in both plants and animals, as we have known. So most of these plants contains the nitrogen from the soil, that is going to be present in the form of the nitrite, or in the form of nitrate or the ammonium ions, but it is very limited. So where the atmosphere consists almost 78% of the molecular nitrogen, but the plants which are unable to convert this particular molecular nitrogen into a useful form because of the lack of the enzyme nitrogenous complex or the nitrogenous enzyme, which is required mainly for the conversion. So only prokaryotic species which possess this particular nitrogenous enzyme. So the nitrogen fixation, which is going to be the first step of this nitrogen cycle, where the molecular nitrogen which is going to be reduced by the nitrogen fixing bacteria to yield the ammonia molecule. So this cycle, which includes three processes, that is ammonification, nitrification and denitrification. If you see in this slide, particular slide, so how this nitrogen cycle which is going to be happening in the nature. So you can see here, so nitrogen which is present, almost 70% of the nitrogen which is present in the atmosphere, which will be undergoing the nitrification. Through the nitrification process, it will be from the atmosphere and as well as the industrial what area. So where you can see this particular process of this, through this process of this nitrification process, it is converting nitrite to the nitrate, that nitrate form. So that nitrates to the nitrate form, which is going to be undergoing nitrification process and entering into the nitrogen to the atmosphere back. So you can see here, through the nitrogen fixation, where the protoplasm of the green plants which are going to be undergoing this particular type of the fixing the nitrogen from the atmosphere, then this atmosphere, this particular plants protoplasm from the from the green plants, which will be utilized by the animals, that is the protoplasm of the animals, so which can enter into the protoplasm of the animals, then it is going to be converting into ammonia through the ammonification process. Then this ammonia, which will be converting into nitrite ion, then this nitrite ion which will be getting reduced to nitrates, then it will be undergoing denitrification process and entering into the atmospheric nitrogen in the form of nitrogen, which is going to enter into the atmosphere through the process of denitrification. So this nitrogen fixation, which is going to happen in the two process, that is types of nitrogen fixation, where the process of the reducing dinitrogen in two ammonia molecules, so that the plants which can absorb the nitrogen is known as nitrogen fixation. So the particular dinitrogen which is getting reduced to ammonia, so the plants directly can now they are having the capacity to absorb this nitrogen through this fixation process, nitrogen fixation process. There are two types, biological and non-biological, symbiotic, under that symbiotic, asymbiotic process. So what is going to happen in the non-biological nitrogen fixation? Let us see. So this nitrogen, first it is going to be fixed by the electrical discharge of the lightning, which is going to happen in the atmosphere. So you can make out here nitrogen, that is N2 plus O2, which is combining gives rise to the nitrous oxide, that is 2NO. Then this nitrous oxide which is going to be formed is combining with again oxygen molecule to form nitric oxide, that is 2NO2. So this nitric oxide which is going to be readily dissolved in the water in the water to form a nitric acid, that is HNO3, and nitrous acids, that is HNO2. So these acids, that is nitric acid and nitrous acids, which are going to be readily releasing the hydrogen, forming the nitrate ions and the nitrite ions. Both ions will be released from this nitric acid and nitrous acids, that is nitrate and nitrite ions. So these nitrate which can be readily utilized by the plants and the microorganisms. You can see here. So HNO3 which is getting undergoing what is that? It is dissociating into H plus and NO3 minus. So this nitrate ions which will be utilized by the plants and the microorganisms, that is HNO2, which will be dissociated into H plus and NO2 minus. So you NO2 plus, you can see here the nitrate ions which can be how the nitrate ions which are going to be formed, which will be utilized by the plants and the microorganisms through this non-biological nitrogen fixation, that is without the involvement of any bacteria or the any organism. So it is undergoing fixation. The particular nitrogen is undergoing fixation mechanism through this process, that is non-biological, without the involvement of any organisms or the bacteria. Now you can see the biological nitrogen fixation, how it is going to happen. So this is it's a prokaryotic domain where because of the presence of this nitrogenous complex enzyme or nitrogenous enzyme, this process is carried out through biological way, where the nitrogen is going to be fixed. So the prokaryotes are going to fix the nitrogen. They are called as nitrogen fixers. So it includes both free living and as well as symbiotic association with the plants. So diazotropes. So mainly the diazotropes, certain strains of this bacteria of the genus Rhizobium, which is going to show the symbiotic association with the legumes of the root nodules. So this symbiotic association which will be done with the legumes via the root nodules. So these species are able to convert molecular nitrogen into ammonia. How this species are going to convert molecular nitrogen into ammonia? So you can see here nitrogen which is utilized, so which is undergoing dissolved in reacting with the water molecule with the involvement of what is that, H plus ions and the 8 electrons which are involved in the reduction process of this particular nitrogenase enzyme. So this converts nitrite, that is N2 nitrogen to ammonia molecule NH3 and H2 plus H2 and ADP molecule is going to be released. So ammonia is going to be formed, which is incorporated either to the glutamate by the glutamate dehydrogenase activity enzyme. So that the glutamate dehydrogenase enzyme which is involved in the amino acid, that is conversion of glutamate or glutamine to alpha ketoglutarate by the enzyme glutamine synthetase, which is getting converted to glutamine. Glutamate is getting converted to glutamine by the enzyme glutamine synthetase or glutamate by the action of the glutamate dehydrogenase, it is getting converted to alpha ketoglutarate. So you have come across in the amino acid metabolism. So this is very important process, biological nitrogen fixation, which was happened with the help of this fixers where it is going to fix the nitrogen molecule through the biological way. So the basic requirements which are required for this nitrogen fixation to carry out. So basic requirements which are required are nitrogenase complex enzyme, as we have seen, the protective mechanism against oxygen, that is leg hemoglobin is required, ferredoxin is required, that is flavored oxygen also we can call it as, and hydrogen releasing system or the electron donor, that is pyruvic acid molecule, and constant supply of the ATP is must, and coenzymes and cofactors like coenzyme A, inorganic phosphate, as well as magnesium ions are required, and cobalt and molybdenum also required for the nitrogen fixation to carry out by the enzyme nitrogenase complex. So you can see biological nitrogen fixation which is going to be carried out by a highly conserved complex of proteins which are called as nitrogenase complex. So this nitrogenase complex enzyme which consists of proteins, many proteins which are mainly required for the fixation process. So which is mainly consists of two important proteins, that is especially iron protein, that is Fe, dinitrogenase reductase. So and another is molybdenum Fe protein, that is dinitrogenase. So the structure is nothing but how this particular nitrogenous complex enzyme structure will be. So this dinitrogenous reductase, which is a dimer of two identical subunits, it is having, and it contains single iron 4Fe4S clusters, redox centers, which are bound between these particular two subunits, and this can be oxidized and reduced by one electron. This dinitrogenous, which is a tetramer with a two copies of two different subunits. So dinitrogenous reductase is different structure, having different structure, whereas dinitrogenous, coming for this dinitrogenous structure, it is a tetramer with a where dinitrogenous reductase is a dimer with a two identical subunits, whereas dinitrogenous enzyme is having a that dinitrogenous alone is having a tetramer with the two copies of two different subunits, which is called as heterodimer, which contains both iron and molybdenum. So you can see its redox centers, which is having two molybdenum subunits and 32 iron clusters, and 30 subunits per tetramer will be present, and it has a two binding sites for this reductase activity, whereas about half of the iron and sulfur is which is going to be present between two bridge pairs of the 4 iron and 4s clusters called centers called as the p clusters. So this p clusters consists of two 4Fe4s clusters linked through additional sulfide ion. So this sulfide ion, additional sulfide ion will be present in this particular p cluster, which we usually having two 4Fe4s clusters. So you can see the action of nitrogenous enzyme, how the action is going to happen here. So you can see in this slide. So this for reducing this nitrogen into ammonia form, this particular nitrogen is which requires the 8 electrons for this activity. So first what it does, dinitrogenous is going to be reduced by transferring of electrons to the dinitrogenous reductase enzyme. So it is first transferring dinitrogenous first is going to be reduced by transferring electrons, 8 electrons to the dinitrogenous reductase, and then this dinitrogenous has the two binding sites for this reductase enzyme. So where the 8 electrons are going to be further transferred from reductase to the dinitrogenous one at a time. So one at a time it is going to transfer from the reductase to the dinitrogenous. Then this reduced reductase binds to this dinitrogenous and transfers single electron, which is oxidized form, dissociates in a repeating cycle. So you can make out in each cycle it requires the hydrolysis of ATP molecule by this dinitrogenous reductase activity. So immediate source of this electrons to reduce this reductase activity is reduced ferredoxine. We can also call it as flavodoxine, right? So this reduced ferredoxine or the flavodoxine, which is mainly required as the immediate source of electrons for the reduced reductase molecule. So ultimate source of the electron to the reduced ferredoxine will be the pyruvate molecule. So pyruvic acid is the ultimate source of the electrons for this reduced ferredoxine, which is mainly required to carry out this particular mechanism of this nitrogenous complex enzyme. So coming for this symbiotic nitrogen fixation, how it is going to happen. So this through the biological way, this symbiotic fixation is going to happen. So symbiotic nitrogen fixation, as I told you in the last slides, which mainly occurs in the plants which harbors this particular nitrogen fixing bacteria within their tissues. So you can see here in the root nodule how this formation is going to happen here. So the best studied example is of the symbiotic nitrogen fixation or the association between the roots of legumes and bacteria of the genus Rhizobium. You can see the Rhizobium bacteria, how the fixation or the symbiotic association is going to happen. It is going to helpful in the roots of nodules of the legumes, which will carry out this symbiotic nitrogen fixation. So the association mainly results from the root nodules in the legumes, where this root nodules is enlarged multicellular structures on the roots which will be present. So you can see in this slide, and legumes will be the particular legumes will be formed by the Rhizobium association, which will fix 25 to 60 kilogram of the molecular nitrogen annually. So you can see, you can make out this root nodule formation here. So what is going to happen, how this root nodule formation is going to happen. First the plant roots which is going to emit flavonoids, that is particularly required for this activity of this nitrogenous enzyme complex, so which will further bind to receptor molecules in plasma membrane. which will be further resulting in the particular, which will be further compatible for the soil rhizobia. So which will help in this, you can make out here flavonoids and rhizobia and the nod factors, right? So in response to these flavonoids, what is going to happen here? The rhizobia, which will be, you can make out here, secretes the nod factors. So that particular nod factors which will go and bind to the receptors in the membranes of the host plant, which will, host plant root, mainly the particular hair cells, right? You can make out here. And then what is going to happen further, these receptor binding, which will cause entry of this calcium ions into the root hair cells, which will further causes this root hairs to swell at their particular tips, and it will, you can make out these tips, right? Tips, and it will curl around the rhizobia, like a rhizobia it will be showing curling, right? So then you can see this infection thread right here. Rhizobia inject the infection proteins to that particular, what is that induced plant roots to develop the infection threads. So you can make out this infection threads where the rhizobia, which is going to penetrate into the root cortex cells. So these proteins, which are known as the nodulins, now this infection thread which is producing this particular what is that protein, that is called as nodulins, which causes root cortex cells to divide now. So that results in forming the nodule. So you can make out the nodules formation, right? Developing of the nodules after the division of this particular root cortex cells after the infections which has happened. So it is going to after the formation of the nodules, so rhizobia invade the nodule cells, now inducing further nodule development. You can make out the further nodule development also. Rhizobia, which is going to divide, then transfers into the bacteroids. Further it is going to transfer into the bacteroids. Now the next process will be nodule becomes pink. You can make out here pink nodules formation inside, as oxygen is regulating the leg hemoglobin which is going to be produced. So you can see this leg hemoglobin which is going to be produced inside this particular pink nodule, right? So this nodule development vascular tissue is that further it is helpful in the further transports the nitrogen compounds to the shoot and organic carbon, and it forms the shoot to the nodule bacteroid. So you can make out this particular process which is happening, right? It is going to transfer to the vascular tissues, right? So it is going to transfer further to the shoot and the organic carbon from the shoot to the nodule bacteroids. You can see the process. So what is happening here, right? Nodule vascular tissue. This is how this process is going to happen, the root nodule formation. So coming for the asymbiotic nitrogen fixation. So this is the last topic of this fixation mechanism. So the free living, as I said you, free living nitrogen fixing organisms which are called as asymbiotic organisms, right? So it includes mainly the aerobic bacteria, anaerobic bacteria, and even blue-green algae bacterial types which may come across like Azotobacter, free-living bacteria, aerobic, those are, and anaerobic free-living which is called as Clostridium, and blue-green algae types which includes Oscillatoria, that is a filamentous, non-heterocystous, that is, and heterocystous if you're coming for the heterocystous filamentous type, that is Nostoc and Anabina. So these are all the symbiotic nitrogen fixation bacteria which are involved in the fixing the free-living nitrogen molecule by through this process, that is asymbiotic nitrogen fixation process. Thank you. In the next slide we will see with respect to the NIF genes.